In a previous lesson on photosynthesis, we learned about the basic photosynthesis equation, where we have reactants on the left side, the carbon dioxide and water molecules, being used to construct one molecule of glucose with 6 molecules of oxygen as a waste. These would be the products on the right. And we learned that the conservation of matter applies here, that during chemical reactions, atoms are not created or destroyed, they're simply rearranged to form new molecules. Well, that process of breaking chemical bonds and making chemical bonds requires energy, and plants are using sunlight as the source of energy to power this chemistry. So in this lesson, we'll take a look a little bit more about the role of light in the process. We'll use our aquatic plant to help us think about this. Being photosynthetic, the aquatic plant needs a source of solar energy to power the photosynthesis chemistry, but it doesn't have to be the sun. It could be this lamp over here, right? So we all know that we can grow lamps indoors using a white light to replace the sun. Well, why is that? And it raises the question, what is it about sunlight that the plants are using? Well, you may be familiar that sunlight consists of all the colors of the spectrum, and we can see this when we use an instrument to separate sunlight into the colors that we see during a rainbow, for example. So here we have the sun here, and this is an instrument called a spectroscope, and if you point this instrument, there's a hole here, at a source of light, it will separate whatever source of light that is, it will separate whatever colors are present into different bands. Now, you shouldn't point this at the sun, but for example, indoors, if you were pointed at one of the ceiling lights, and it's a white ceiling light, you would see the colors of the spectrum. So the white light from an indoor light consists of a mixture of these different colors. Now, what are colors? Well, colors are different wavelengths of electromagnetic radiation. So we can think of light as a wave, and there are different wavelengths for these different colors. A wavelength would be the distance between the crest of one wave and the crest of the next one. So red light has a different wavelength than blue light. And these instruments, whether it be a spectroscope or a prism, can separate light into the different wavelengths that are present in the original light source. Well, it raises a question, are all of the colors of light necessary for photosynthesis? If we think that, well, plants need sunlight to grow, well, do they need all of the colors that are present in sunlight? Well, there's an easy way to figure this out. What you could do, back to our aquatic plant, is you could just set up different aquatic plants and then shine different colors of light on the plants. So some plants you might have red light, some plants you might have green light, some plants you might have blue light shining on them, and then you could simply measure the rate of photosynthesis. Now, often the way scientists measure the rate of photosynthesis, you can see little bubbles here, is to simply measure the production of oxygen bubbles. Remember, oxygen is a waste product of photosynthesis, so it's a good measure of the production of glucose. And here we actually see some bubbles being produced. This aquatic plant was illuminated by white light, as it turns out, and we see lots of oxygen bubbles. So we're going to use oxygen production as a measure of photosynthesis, and we'll simply see if there's any difference when we shine different colors of light on the plant. The results can be seen in this graph. Notice we have on the x-axis the wavelength, so this would be the color. And we have on the y-axis the relative rate of photosynthesis as measured by oxygen production. That's an easy way to measure photosynthesis. And notice then, here's the data. When you shine blue light on a plant, you get relatively high rates of photosynthesis or oxygen production. When you shine green or yellow, you get significantly lower rates of photosynthesis. When you shine red light, you get a peak again. Get higher rates of photosynthesis. So it looks like not all colors are equal here in driving photosynthesis. You get maximum photosynthesis when you shine blue light or red light, minimum photosynthesis when you shine green or yellow light. Well, that raises a question, why is this? Well, the answer to that question has to do with how chloroplasts, the site of photosynthesis, how chloroplasts interact with light. Now, scientists have learned, remember the chloroplasts are the green structures here, scientists have learned that on the membrane of the chloroplast, all around the membrane, inserted into the membrane, are very important molecules called chlorophyll. Chlorophyll molecules are pigments. These and a pigment would be a molecule that interacts with light in a certain way. So chlorophyll, being a pigment, it can absorb certain colors of light and it reflects other colors of light. So these chlorophyll molecules are the important molecules that are interacting with light, and they exist on the surface of each chloroplast. So to study how the chlorophyll molecules interact with light, we're going to do a procedure to extract these chlorophyll molecules, to rip them off of the membranes here, to take them out of their membranes and put them in a solution. Then we can study how chlorophyll interacts with light. The first step is to take a leaf, a green leaf, and start boiling it in alcohol. And here we see we're looking into the beaker and the leaves are boiling. Now what happens over time is that the leaf starts to lose its green color. It starts to sort of get pale, starts to turn whitish, and then the alcohol starts to turn green. What's happening is the chlorophyll molecules are being extracted from the membrane of the chloroplast, and now the molecules are floating around in the alcohol solution. And so we can get a test tube filled with chlorophyll molecules in alcohol. Now we can use this test tube to examine the absorption properties of chlorophyll. And here's how you do that. Remember, you take your spectroscope. Remember, that's a device that can separate whatever the light source is into the colors that are present. So for example, if it were sunlight, the spectroscope would show us the full spectrum, and we see that in the picture up here. Here's the hole, the opening of the spectroscope that would be pointed to the light. And then when you kind of look to the left or the right, you'll see whatever colors of light are present in the light source. So for sunlight, we see the entire spectrum. We see the reds and the greens and the blues. We're not showing all the other colors here, just for simplicity. Now what we do is we take our test tube filled with chlorophyll. We have sunlight here, but we put the test tube in between the light source and the spectroscope, and notice what has happened here. What we see is we see the green band. So here's the light source, here's the chlorophyll. You can kind of see the test tube is in front of the light source, and here is the only band that we really see is a green band here. The blues and the reds have disappeared. We don't see the blue bands or the red band here. So the blues and the reds have disappeared. Well, now why did the blues and the reds disappear? And scientists explain this by saying what's happening is the chlorophyll molecules are absorbing the blues and the reds, but they're reflecting the greens. So the reflected green light can keep on moving through the spectroscope and end up in our eye as we look through the spectroscope. So what chlorophyll molecules do is apparently they absorb the blues and the reds preferentially. They do not absorb the greens. We can see this in another graph. On the x-axis, again, we have wavelength, so this would be the different colors of light. And here on the y-axis, we have estimated absorption, so the percentage of the light that is absorbed at that color of light. Notice there happen to be two types of chlorophyll, A and B. Don't worry about that, but notice where the peaks are. The peaks are in the violet and blue region here, and in the red region. In other words, the chlorophyll molecules, those molecules that are on the surface of the membrane of the chloroplast, those molecules absorb blue light, they absorb red light, but they do not absorb the green and yellows. Now, this is important because the absorbed light then would be energy that is being captured by the chloroplast to power chemistry, right? The green light that gets reflected is why the chloroplasts appear green in the first place, right? So now we're starting to understand a little bit of the physics here of why is it that plants are green? Well, the cells have these green discs. Why are the chloroplasts green? Well, because chlorophyll molecules on the surface are reflecting green light but absorbing the blues and the reds. And we can see that in this diagram as well. Here we have sunlight with all the colors, but the red, green, blue are the only ones pictured here, striking the leaf. The blues and the reds will be absorbed, and that will be the energy to power photosynthesis, while the green light is reflected. Whatever the light that's reflected, that's the color that the object will appear to us. So let's now take these two graphs and see how they match up, how they stack up next to each other. Here we have the absorption graph. So this tells us that chlorophyll molecules absorb the blues and the reds. And remember the first graph we looked at, the rate of photosynthesis. Oh, take a look. We get a peak in the blue and a peak in the red. And that's no accident. The reason why plants grow best under blue or red is because plants can absorb the blue and the red light. The reason why plants do not grow well with green and yellow light is because plants reflect. The chlorophyll reflects the green and yellow. The light that gets reflected is not available to power the chemistry of photosynthesis. It's the absorbed light, the blues and the red. That's the source of energy to power the chemistry of photosynthesis. So when we say light here, typically out in the world, of course out in nature, it's just sunlight hitting planet Earth here. But specifically, due to the presence of chlorophyll, it's the blues and the reds that are the valuable colors of light for this particular process of photosynthesis. Okay, let's summarize then the role of light in photosynthesis. We know that for plants to make sugar, they're going to have to break the chemical bonds of the starting materials, the carbon dioxide and water molecules, rearrange the atoms, and make new chemical bonds to form glucose with some oxygen as waste. Well, that chemistry requires energy, and plants are getting that energy from the sunlight. But now we know from examining the properties of chlorophyll molecules that are on the surface of chloroplasts that the light coming from the sun is not all equally valuable. The green light in the sunlight is being reflected. This is why the chloroplasts appear green. It's the blues and the reds that are being absorbed. Absorbed sunlight energy is the captured energy to power the process of photosynthesis.